Contraction of muscles. Let's see the structure of skeletal muscle. Skeletal muscle is made up of large number of fasciculi which are wrapped by connective tissue sheet called as epimysium. Each fasciculus is further covered by perimysium and is made up of muscular fibers, that is myofibers. Each muscle fiber is lined by plasma membrane called as sarcolemma, which encloses sarcoplasm. Muscle fiber is syncytium, as sarcoplasm contains many nuclei. Endoplasmic reticulum, that is sarcoplasmic reticulum of the muscle fiber, stores calcium ions, which are released during action. A characteristic feature of the muscle fiber is the presence of large number of parallelly arranged filament in the sarcoplasm called myofilament or myofibrils. Each myofibril has alternate dark and light bands. Light bands contain actin and is called I band or isotropic band, whereas dark bands called A or anisotropic band contains myosin. Both the proteins actin and myosin are arranged as rod-like structure parallel to each other and also to the longitudinal axis of the myofibrils. Actin filament are thinner as compared to myosin filament, hence are commonly called thin filament. Myosin are thick filament. In the center of each I band is an elastic fiber called Z-line which divides I band. Thick filament in the A band are also held together in the middle of this band by thin fibrous membrane called M-line. The A and I band are arranged alternately throughout the length of the myofibrils. The portion of the myofibril between two successive Z-line is considered as the functional unit of contraction and is called sarcomere. So the region between two Z-line is called sarcomere. Now let's study about actin and myosin. Actin is thin, it's a complex type of contractile protein and it has three different components. That is F-filamentous actin, tropomyosin and troponin. F-filamentous actin, it's a backbone of actin filament. Each F-actin is a polymer of monomeric G-actin, that is globular actin. So these are globular actin. Two F-actin are wound to each other. It is a double-stranded tropomyosin. Two filament of tropomyosin also runs close to F-actin throughout its length. Each tropomyosin is loosely attached to F-actin. In resting stage, that is during relaxation, tropomyosin physically covers the active binding site for myosin of the actin strand. Troponin, it is a complex of three globular protein. It is distributed at regular interval on tropomyosin. In the resting stage, that is during relaxation, a subunit of troponin marks cover the active binding site for myosin on the actin filament. It has affinity for actin, tropomyosin and calcium ions. Myosin, it's a thick dark band. Each myosin filament, thick filament, is a polymerized protein. Many monomeric proteins called myromyosin constitute one thick filament. Myosin molecule consists of two heavy chains coiled around each other. Each myromyosin consists of two important parts: a globular head with a short arm, that is heavy myromyosin HMM, and a tail, that is light myromyosin LMM. The HMM component, that is head and short arm, projects outwards and is known as cross arm. The globular head is an active ATPase enzyme and has binding site for ATP and active site for actin. It splits ATP to produce energy. In the center of sarcomere, myosin head and actin are absent. Now let's see the mechanism of muscle contraction. It is explained by sliding filament theory, which states that contraction of a muscle fiber takes place by sliding of thin filament over the thick filament. So you can see here, means here the actin slides over the myosin. Muscle contraction is initiated by a signal sent by the central nervous system via motor neuron. A motor neuron along with muscle fiber connected constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called neuromuscular junction. So this is neuromuscular junction or motor end plate. A neural signal reaching this junction releases neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. This spread through the muscle fiber and cause the release of calcium ions in the sarcoplasm. These calcium ions are released from sarcoplasmic reticulum. Increase in calcium level leads to the binding of calcium with the subunit of troponin and actin filament, and thereby removes masking of active site for myosin. So this region becomes uncovered. Utilizing the energy from ATP hydrolysis, the myosin head now binds to the exposed site on actin to form cross bridge. This pulls the attached actin filament toward the center of A band. So you can see over here, the Z line attached to this actin are pulled inwards, thereby causing a shortening of the sarcomere, that is contraction. So it is clear that during shortening of muscle, that is contraction, I bands get reduced, whereas A band retain the length. A new ATP binds and the cross bridge is broken. The ATP is again hydrolyzed by the myosin head, and the cycle of cross bridge formation and breakage is repeated, causing further sliding. The process continues till the calcium ion are pumped back to the sarcoplasmic cisternae, returning in the masking of actin filament. So when calcium ions are removed, the myosin binding site on actin is again is covered. This causes the return of Z line back to their original position, that is relaxation